Hello students, hope all of you are fine. So dear children, we are doing the lesson Cell, the basic unit of life, and this is your last video of the lesson. Okay, in this video we are going to discuss about the nucleus and chromosomes. Okay, nucleus. So what is nucleus? Nucleus is a specialized double membrane bound protoplasmic body. Okay, and it contains all the genetic information for controlling cellular mechanism and transmission to the posterity. Okay, the nucleus is the non dividing or metabolic phase is called the interphase. Okay, if a nucleus is not dividing, then we say that it is in interphase. Okay, then like other cellular or structures, living sustained nucleus does not show much internal differentiation. For detailed study of the nucleus, the cell must be properly killed, fixed and stained. Okay. Now nucleus is the largest cell organelle. Though first observed by Leeuwenhoek in the red blood corpuscles of the fish, nucleus was first studied in orchid root cells by Robert Brown in 1831. A nucleus is present in all living eukaryotic cells with the exception of mature sieve cells of vascular plants and red blood corpuscles of the mammals. Even here, a nucleus is present during the early stage of their development. Presence of hereditary information in the nucleus was proved by the work of Joachim Hammerling, okay, in 1953 on single-celled algae, that is Acetobularia. Okay. Number commonly cell are uninucleate, that is they possess a single nucleus. The Protistean paramecium chordatum has two nuclei, okay, that is binucleate, macronucleus for controlling the metabolic activities of the organism and micronucleus possessing hereditary information. The Protistean paramecium chordatum, as I told you, it has two nucleus. Multinucleate are the polynucleate condition is found in some of the cells of the bone marrow, striated muscle, latex vessels, several fungi and algae. Multinucleate animals or the Protistean cells are called syncytial cells, epidermis of ascaris, while in the plant and fungi they are called cynocytic cells. Okay, for example, rhizopus, wucheria. Okay, acellular slim molds have the multinucleate protoplasmic body called the plasmodium. Position: nucleus is usually found in the region of maximum metabolic activity in the cytoplasm. Commonly, it is situated in the geometric center of the cell. In plant cell, it is pushed to the peripheral position on one side due to the development of large central vacuole. Nucleus is peripheral in fat storing cell or adipocytes. The basal in the glandular cell. It is suspended in the central vacuole by the cytoplasmic strands in the spirogyra. If we go to the shape of the nuclei are generally rounded in outline. They appear oval or elliptical in the plant cells, having large central vacuole. Disc-shaped nuclei occur in the cell of the squamous epithelium, lobed in white blood corpuscles, and irregularly branched in the silk spinning cells of the insect. Now, biochemical analysis of the DNA or the nucleus. DNA 9 to 12%, RNA 5%, lipids 3%, basic proteins 15%, acid protein, neutral protein and enzymes constitute 65%. Traces of minerals like calcium, magnesium, potassium and sodium. Phosphorus is constituent of DNA and RNA, and acid proteins. Okay, so this is the chemical analysis of the nucleus. Now let's see the ultrastructure of the nucleus. A typical interphase nucleus is 5 to 25 micrometer in diameter. It is differentiated into 5 parts: nuclear envelope, nucleoplasm, nuclear matrix, chromatin, and nucleolus. Now, nuclear envelope. Karyotheca. Okay, nuclear envelope is also known as karyotheca. It bounds the nucleus on the outside. The nuclear envelope separates the nucleus from the cytoplasm. It is made up of two lipoproteins and trilaminar membrane, each of which is 60 to 90 Armstrong thick. The inner membrane is smooth. The outer membrane may be smooth, or its cytoplasmic surface may bear ribosomes like the rough endoplasmic reticulum. The two membranes of nuclear envelope are separated by an electron transport perinuclear space. The space is 100 to 500 Armstrong in the width. The outer membrane is often connected to the endoplasmic reticulum. Nuclear envelope contain a large number of pores or perforations. In some cases, 10% of the envelope is occupied by the pores, and two membrane of the envelope become continuous in the region of the pores. Nuclear pore have complex structure. They may have diaphragm, septum, plug of the electron dense material, or nucleoplasmin, blebs, or annuli. Annuli are circular structures around the pores. The pores and their annuli form pore complex called annulated pores. An annulated nuclear pore may possess 9 cylinders, a central and 8 peripheral. Instead, they may be a network of granules and the filament. The nuclear pores control the passage of substances to the inside or outside of the nucleus. For example, RNAs, ribosomes, and proteins. Now next is nucleoplasm. So nuclear sap or karyolymph or Strasburger, okay, these are the terms which are used for nucleoplasm, and these terms they were given in 1882. Okay, it is transparent, semi-fluid, and colloidal substance which fills the nucleus. It contains nucleoside and number of enzymes. For example, DNA polymerase, RNA polymerase, nucleoside, and phosphorylase. Okay, nucleoside phosphorylase, which are required for the synthesis and functioning of DNA, RNA, nucleoproteins, etc. Some of the proteins present in the nuclear plasm are essential for spindle formation. Now next we have is the nuclear matrix. It is the network of fine fibrils, okay, of the acid protein that function as the scaffold for the chromatin. Okay, on the periphery below the nuclear envelope, nuclear matrix form a dense fibrous layer called the nuclear lamina. Okay, terminal end of the chromatin fiber or telomeres are embedded in the nuclear or the fibrous lamina. Nucleus matrix consists of two types of intermediate filament, lamina A and lamina B. The nuclear matrix and the nuclear lamina form scaffold for the chromatin, attachment site for telomeric parts, mechanical strength to the nuclear envelope, and component of nuclear envelope complex. Next we have is the chromatin. It is the heredity DNA protein fibrillar complex, which is named so because of its ability to get stained with certain basic dyes. Okay, in Greek the chroma means colored and flaming. Okay, Uh, it was given by Fleming in 1879. Chromatin occurs in the form of fine overlapping and coiled fiber which appear to be produced as a network called the chromatin reticulum. Chromatin fiber are distributed throughout the nucleoplasm. They are differentiated into two regions, euchromatin and heterochromatin. Okay, Hinch, okay, in 1928. Euchromatin is narrow, 10 to 30 nanometer thick, lightly stained and diffuses, okay, and it is diffused fibrous part which forms the bulk of the chromatin. Heterochromatin is wider, 100 nanometer thick, darkly stained and condensed granular part which is attached here and there on the euchromatin. Depending upon the size of the granules formed by the heterochromatin, they are called chromatocenters, karyosomes, or false nucleoli. The whole of the chromatin is not functional. Generally, only a portion of euchromatin which is associated with acid protein take part in the transcription or formation of the RNAs. During prophase of nuclear division, the chromatin fiber condense to form a definite number of thread-like structures called the chromosomes. Now next we have is the nucleolus. Now nucleolus, okay, if you see, it was first discovered by Fontana in 1781, and it was described by Wagner in 1840. Okay, so and provided with its present name by the Baumann in 1840. So three scientists are involved. First, in 1781, Fontana, okay, he discovered, and Wagner, he described it in 1841, and Baumann in 1840 gave the name nucleolus to it. Nucleolus is a naked, round, or slightly irregular structure, okay, which is attached to the chromatin at specific region called nucleolar organizer region, okay, that is NOR, nucleolar organizer region. Commonly 1 to 4 nucleoli are found in the nucleus, okay. The number of nucleoli can be 1 to 4, okay. Up to 1600 nucleoli are present in the oocyte of xenopus, okay. If you see the xenopus, about 1600 nucleoli are present in single xenopus. A covering membrane is absent around the nucleolus. Because of this, the content of the nucleolus are in direct contact with the nucleoplasm. However, calcium seems to help in maintaining its configuration. Nucleolus has 4 components: amorphous matrix, granular parts, fibrillar portion, and chromatin, okay. Amorphous matrix, it is homogeneous ground substance of the nucleolus. Matrix is formed of the protein, okay. The granular portion, it consists of granular or granules of size 150 to 200 Armstrong which lies scattered in the amorphous matrix. The granules are formed of protein and RNA in the ratio 2:1. They are believed to be precursors of the ribosomes. Fibrillar portions, or that is also known as nucleolonema, okay, nucleolonema. It is formed of large number of small fibrils that are 50 to 80 Armstrong long. The fibrils are made of both protein and RNA and are believed to be the precursor of the granules. Now next is chromatin portion. It is part of chromatin which is associated with nucleolus. Depending upon its position, nucleolar chromatin is of 2 types: perinucleolar and the intranucleolar. The perinuclear chromatin lies around the periphery of the nucleolus. It gives rise to ingrowth or the trabeculae, which produce the intranuclear chromatin. The nucleus is principal site for development of ribosomal RNAs. It is center for formation of ribosome component. Nucleus stores the nucleoprotein. The same are synthesized in the cytoplasm over the ribosome and transferred to the nucleolus. It is essential for spindle formation during the nuclear division. Now, function of the nucleus. Nucleus is essential and integral part of eukaryotic cells. It stores genetic information in its DNA molecules, which can be passed on to the daughter cell. It also controls cellular activities. Chromatin. Nucleus contain heredity material called chromatin. Chromatin is DNA protein complex, okay, that is the combination of DNA and protein. It is made of number of fine fibers that condense to form the chromosome. Number of chromosome is fixed for a species. They bear genes. Genetic information. Chromatin part of nucleus possesses all the genetic information that is required for growth and development of the organism, its reproduction, metabolism and behavior. Cellular activities. Nucleus controls cell metabolism and other activities through the formation of RNAs, that is messenger RNA, then ribosomal RNA, transfer RNA, which control synthesis of particular type of enzyme. Ribosomes are formed in the nucleolus part of the nucleus. Variation. All variations are caused by change in the genetic material present in the nucleus. Cell growth and the maintenance. With the help of RNA, the nucleus directs the synthesis of some structural proteins and chemicals required for cell growth and maintenance. The cell differentiation. It directs cell differentiation by allowing certain particular sets of genes to operate. Now, next is cell replication. Replication of the nucleus is essential for the cell replication. So with this we complete the nucleus, and now let's do the chromosomes. Okay, so chromosomes, they are rod shaped or thread like, deeply stainable condensed chromatin fibers which are hereditary vehicles as they store and transmit coded hereditary information. Now, chromosomes appear only during karyokinesis, that is the division of nucleus. They are meant for the equitable distribution of genetic material. Number is fixed and is the same in all the individuals of the species. There is a single set of gametophytic or haploid form and two set of saprophytic or diploid forms. Okay, size and shape of the individual chromosomes are quite distinct. The shape is more clearly visible in late prophase and metaphase as well as anaphase. When primary constriction or the centromere become distinct during prophase and metaphase, the chromosomes are replicated. There are two chromosome halves or the chromatids. The two chromatids are attached to each other by a narrow area called the centromere or primary constriction. Anaphasic chromosomes do not have the chromatids. The two part of the chromosomes or chromatid on either side of the primary constriction are called the arms. Okay, the two arms are equal in isobranchial chromosome and unequal in the heterobranchial chromosome. The ratio between the two arms of the chromosome is called the centromeric ratio. Based on the position of centromere, chromosomes are of four types. Now, first is telocentric, you can see. Okay, the centromere terminal in area of telomere. Next is acrocentric, the centromere inner to the telomere, that is subterminal. Okay, then we have submetacentric. Okay, third one you can see the centromere is submedian, and next is metacentric, that is the centromere is in middle. Beside primary constriction or the centromere, a chromosome may have one or more secondary constriction. A secondary constriction present near the distal part of an arm may develop a small outgrowth or the fragment called the satellite. A satellite is connected to the secondary constriction through a chromatin thread. The chromosome having satellite is called the set chromosome. Set chromosome are called marker chromosomes. Other secondary constriction can also be also function as the marker because they occupy a constant position. Under light microscope, the cytologists found that the centromere contain a coiled filament called the chromonema. Okay, chromonema was thought to be gene bearing part. Okay, some worker thought that a chromosome may have several chromonemata. Electron microscope has revealed that a chromosome is actually formed by direct condensation of the loops of the chromatin fiber attached to a scaffold. It is 30 nanometer in diameter and contains a single DNA duplex. Now next we have is the giant chromosome. There are of two types, that is polyteny and lamprous chromosomes. Now polyteny chromosomes, and polys means many, tinea means thread. Okay, so it was given by Collar in 1882. Polyteny chromosomes were first reported by E.G. Balbinia. Okay, Balbini in 1881. They are quite common in the salivary glands of the insects and are therefore popularly called the salivary chromosomes. Polyteny chromosomes also occur in other organs of the insect, antipodal cells, or the embryo of the embryo sac. Okay. Now endosperm cells and the suspensor cells of the embryo. Okay. Then the chromosomes can reach a length of 2000 micrometer and contain 1000, that is Drosophila, to the 16,000 in chironomous times DNA as compared to the ordinary somatic chromosomes. That means the number of chromosome can be 1000 times more, or it can be 16,000 times more than the somatic chromosomes. Okay, that is present in the salivary gland. Polyteny chromosomes are multi-stranded. Okay, they are permanent prophase stage. Okay, the giant chromosome, okay, the giant chromosomes are multi-stranded. They are in permanent prophase stage. Okay, the giant chromosomes are formed by somatic pairing between the homologous chromosomes and repeated replication, that is endomitosis of their chromonemata. Okay. Now, all the polyteny chromosome may remain attached to one another at a common point, and this is called the chromocenter. It represents pericentromeric heterochromatin, which is slow to replicate. Polyteny cells and their nuclei are large sized. Polyteny cell cannot divide further. They ultimately die. The adult organs develop from some small size diploid cell lying nearby. Depending upon their reaction to the basic dyes, the polyteny chromosomes bear number of dark bands of various size and intensity. They are separated by light areas called the interbands. The dark bands are assumed to be formed by the juxtaposition of the chromomeres of the different chromonemata of the polyteny chromosomes. In certain development developmental stage, the polyteny chromosomes bear conspicuous swellings called the chromosome puff. Okay, the larger swelling are called the Balbini rings. In the region of puff of the Balbini ring, the DNA strand uncoil, becomes active and produce number of copies of the messenger RNA. The messenger RNA may remain temporarily stored in the puff. Puffs are not permanent at different physiological or developmental stage. Different bands uncoil to produce puff. Puffs are withdrawn after the completion of the stage. By correlating puff with the different physiological or developmental processes, scientists have been able to locate gene on the polyteny chromosome and prepare chromosome maps. Now, next chromosome that we are going to discuss is the Lamprey's chromosome. Now, Lamprey's chromosomes are highly elongated, special kind of synapsed mid-prophase or diploteny chromosome bivalent, which are already undergone crossing over. They were first seen by Fleming but were described by Rutger in 1892. Lamprey's chromosomes occur in the diploteny stage of most animal oocytes, spermatocytes of many, and even giant nucleus of the unicellular algae, that is the Acetabularia. Okay, and the Lamprey's chromosomes are larger than even polyteny chromosomes. Their total length in urodale oocyte may be up to 5,900 micrometer or 3 times the aggregate length of the total polyteny chromosome. Lamprey's chromosome occur in pair. The pair consists of homologous chromosomes which are joined at certain contact point called the chiasmata. Each chromosome has a double main axis due to presence of two elongated chromatids. Both the chromatids bear rows of large number of chromomeres. Two adjacent centromeres or chromomeres are separated by the interchromomeric sketches. Okay, many of the chromomeres give out lateral projections or the loops. The lateral loop provide a test tube or the Lamprey-like appearance to the chromosome pair. Length of lateral loop may vary from 5 to 100 micrometer. Loops are uncoiled or expanded parts of chromomere with one to several transcriptional units. Usually a lateral loop has a thin or uncoiled part and a thick or coiling part. Lateral loop take part in the rapid transcription of the messenger RNA meant for the synthesis of yolk and other substances required for the growth and development of the myocytes. RNA synthesis begins at the thinner end. It progresses towards the thicker end. The transcript, along with their binding proteins, remain attached to the loop and give it a fine fibrillar appearance. Some messenger RNAs produced by Lamprey's chromosomes may be stored in the informosomes, that is the messenger RNA plus protein, for producing biochemicals during the development of embryo. After full development of myocytes, the lateral loops are withdrawn and the chromosomes get shortened. Now let's discuss the function of the chromosomes. Chromosomes contain genes. Okay, all the hereditary information is located in the genes. Chromosomes control the synthesis of the structural protein and thus help in the cell division and cell growth. They control cellular differentiation by directing the synthesis of the particular enzymes, chromosomes control cell metabolism. Chromosomes can replicate themselves or produce their carbon copies for passage to daughter cells and the next generation. Set or satellite chromosome produce nucleoli for the synthesis of the ribosome. Their haploid or diploid number respectively bring about gametophytic and sporophytic characteristic of to the individual. Chromosomes form a link between the offspring and the parents. Some chromosomes called the sex chromosome, that is X and Y, or X and O, determine the sex of the individual. Though the process of crossing over chromosome, through the process of crossing over chromosomes introduce variations. So variations that we see are because of the crossing over of the chromosomes. Mutation are produced due to change in the genetic chemistry or the chemistry of the genes. Okay, so with this we conclude the lesson.